Okay, so welcome to this next video on smooth muscle contraction. In this video we want to talk about what sort of a calcium signal you're going to get if you just have a local stimulation of acetylcholine. Okay, so we've seen that if you stimulate the entire cell with acetylcholine, then IP3 levels are going to go up within the entire cell, and IP3 going up within the entire cell means that all of the IP3 receptors all along the cell are going to be primed with IP3, i.e. if calcium now binds to them, it's going to have a stimulatory effect. So what this means is that you can get these propagating releases of calcium along the sarcoplasmic reticulum known as a calcium wave. So that's the sort of calcium signal that you can get if you have a global acetylcholine signal. Okay, so now let's look at what you can get if you have just a local acetylcholine signal. So local acetylcholine signal. Right, okay, so this time you're only going to deliver acetylcholine onto a small region of the smooth muscle cell. So let's say this is our smooth muscle cell here. So this time we've only delivered acetylcholine, let's say, onto this small region here. So this region, let's say we deliver acetylcholine onto this region. Okay, again it's going to work through the exact same pathway. It's going to activate the muscarinic receptors of the third type, so the M3 receptors, and it's going to cause IP3 to go up. But this time you're only going to get IP3 going up in this small region around the membrane where you've actually released acetylcholine onto. Okay, now if I draw the sarcoplasmic reticulum again, here's the sarcoplasmic reticulum. This time, it means that not all of the IP3 receptors are going to get IP3 bound to them. Only those IP3 receptors in the vicinity of the rise in IP3, i.e. these IP3 receptors here, are going to get IP3 bound to them, whereas these ones over here, they're not going to have IP3 bound to them. And remember, IP3 receptors which don't have IP3 bound to them have a calcium inhibitory binding site available. So if calcium now binds to these IP3 receptors that aren't in this pink region, what it's going to cause is not the IP3 receptor to open, but the IP3 receptor to be inhibited, so to reduce the chance that the receptor is open further, basically. Okay, now, what's going to happen is when IP3 goes up nearby here, these ones, at least, in this pink region, are going to get IP3 bound. So they're now going to have 4 stimulatory calcium binding sites exposed. Now, something is then going to lead to calcium binding to one of them, let's say. So one of them is just going to happen maybe to get 4 calciums binding to its calcium stimulatory binding sites. So remember, the calcium concentration intracellularly is not 0, basically. It is around 100 nanomolar, so I'll put it up here so that I don't go through the cell membrane. So it's not impossible that one of them will get 4 calciums binding to it with that calcium concentration. Alternatively, there may well be some mechanism by which calcium is going to come through the membrane as well. The acetylcholine might also stimulate an ionic pathway. But something is going to mean that one of these IP3 receptors just happens to get 4 calciums binding to it. Okay, that's going to mean that it releases calcium, basically, and I'm going to ruin the picture if I draw it on that one. So I'll say this one also has IP3 bound to it here, so it's going to release calcium. Okay, now, in this pink region where you have had this local rise in IP3, I have drawn 2 IP3 receptors that are in this pink region, but of course there will be loads, absolutely loads. So there are loads of IP3 receptors in this local pink region which all have IP3 bound to them. Now when this one IP3 receptor opens and allows calcium to leave the sarcoplasmic reticulum and go up in the vicinity of this receptor, then what will happen is that that calcium will spill onto the other primed and ready IP3 receptors, and it will cause them to open. So all these other ones will also open that are in the region where IP3 has gone up and which have IP3 bound. However, when calcium spills out of the pink region and onto the IP3 receptors in the neighbourhood of the pink region, but which don't have IP3 bound, what's going to happen is that calcium is going to now bind to inhibitory calcium binding sites, and is therefore going to make the chance of those IP3 receptors being in the open conformation smaller. So it's going to inhibit these ones. So these ones certainly are not going to now open. So you're not going to get the propagating effect that we saw in the case of a calcium wave. Okay? Instead, what you get is a local rise in calcium as all the IP3 receptors in this local region, this pink region here, all open and release calcium from the sarcoplasmic reticulum. So you're going to get a local rise in calcium, basically. And that sort of a calcium signal is known as a calcium puff. Okay? So calcium puff. Right. So that's what's going to result as a... Well, that's what we're going to get if we have a local acetylcholine signal. We're going to get a local rise in calcium, basically. Okay, so global acetylcholine stimuli give rise to calcium waves across the cell, and the frequency of the wave is then encodes the intensity of the acetylcholine stimulus. Local acetylcholine stimuli give rise to local calcium rises in the vicinity of where the acetylcholine stimulus was given. Okay, right. Now what we want to discuss is how in both of these cases the calcium stimulus is going to cause contraction. Now, in the case of the global calcium stimulus, what you're going to get is a global contraction of the smooth muscle cell. Whereas in the case of this local calcium stimulus, again, you're just going to get local contraction of the smooth muscle cell. In this one here, you will get contraction of the contractile units that are in that region of the smooth muscle cell where calcium has gone up. Whereas in this one up here, you're going to get contraction of the contractile units all over the cell. Okay, right. So, now that we've got that off our chest, we can reconverge both pathways and see how calcium going up is going to activate the contraction of the contractile units, whether that be in a local region or in a global region. Okay, right. So, what happens is that calcium binds to a protein known as calmodulin. So, let's begin our discussion of calmodulin then. So calmodulin is a protein that in cartoons is often drawn like this. It has two lobes, okay, here and here, which are the N lobe, which I'll denote here, and the C lobe, which I'll denote here. Now, both the N lobe and the C lobe have calcium binding sites, so each one has two calcium binding sites. So the overall calmodulin protein has 4 calcium binding sites. Now these calcium binding sites are what are known as EF hand domains. So let me explain to you what an EF hand domain is. Okay, so an EF hand domain is a loop, a polypeptide loop basically, that looks kind of like this. Okay, so it's a domain within a protein that is capable of binding to calcium ions, to divalent calcium ions. Okay, and if this line represents the polypeptide, i.e. represents the string of amino acids following one another, then an EF hand domain is a loop in this polypeptide like so. Now, the amino acids that make up this loop region of this polypeptide, basically they are all acidic residues. So these amino acids will be acidic residues, which means that their R group is an acidic side chain basically, and is capable of donating protons away. So an example basically would be glutamic acid. So let me draw the structure of glutamic acid. So here's the basic amino acid structure. Here's the amino group. Here's the alpha carbon. Below that, the carboxyl group down here, or the carboxylic acid group. The hydrogen off the alpha carbon, and then glutamic acid has effectively a propanoic acid group coming off it like so. So here's glutamic acid. Right, and then just hydrogens off these carbons here, these two methylene groups. Right, so this is the structure of glutamic acid here. And basically, glutamic acid is capable of donating a proton from this hydroxyl group here. So this is the R group of glutamic acid. And it's capable of donating a proton from this hydroxyl group. Now, when it does, what's going to happen to this hydroxyl group? Well, the proton is going to leave. That will have a positive charge. So here's the proton off here. And now that will leave the oxygen basically with a negative charge. So the oxygen will be left with the electron that hydrogen has vacated effectively. Because all that's gone off here is the hydrogen nucleus, the proton. So the electron of the hydrogen has remained with the oxygen. So the point is that once the proton has left the acidic side group, it leaves this acidic side group with a negative charge. This is what is known as the conjugate base of the acid. Okay, so this is the conjugate base of the acid. And the strict name for this molecule now with a negative charge on the oxygen and no proton here is glutamate. Now people often use glutamate and glutamic acid interchangeably, and they've got a fair point for why they're doing that, because in a solution containing glutamic acid you'll have loads of molecules that are actually glutamic acid, i.e. they have the proton on their oxygen, and then you'll have loads of molecules that are glutamate, i.e. they've lost their proton, and it will be in an equilibrium basically. But strictly speaking, glutamic acid is the molecule where you have a hydroxyl group here, and glutamate is the conjugate base of glutamic acid, the molecule when it's lost its proton. Okay, the important thing is that acidic amino acid residues, when they donate their protons away, they get a negative charge. So you're going to have a lot of amino acid residues here which have a negative charge, and those basically will interact quite nicely with the calcium ion in the center, which is a divalent cation, so it has a positive charge. Okay, and that's how these EF hand domains bind to calcium ions. Okay, now EF hand domains don't generally exist on their own. They generally exist in dimers. So generally what you find is EF hand domains next to one another. So you'll have one like this, and it will have a neighbor attached to it. So you often find EF hand domains in dimers. So this is an EF hand dimer, basically. And that means that these EF hand dimers combined two calcium ions overall. So one here and one here. So that's what these two lobes of Calmodulin have, basically. They have these EF hand dimers nets where you can bind two calcium ions. So each of these two lobes of Calmodulin has each lobe of Calmodulin has an EF hand dimer. We'll continue this discussion in the next video.